Thank you, Jay. With this wonderful title, I better get right into it and start explaining what it is that I'm doing with texture analysis on Geyserite deposits. So the first thing, what is a Geyserite? Well, Geyserites are silica deposits that are found around silica depositing hot springs. The top two images are Geyserites. They were collected near the vent of a hot spring in Yellowstone National Park. The one on the left is a spicular Geyserite that formed in the splash zone where wave activity, be it from the surging of the hydrothermal fluid into the vent or wind, splashed water, and it formed spicules, which the image is cropped and so they're cut off a little bit. But the next one on the right there at the top is a subaqueous Geyserite that, as the title implies, it formed under the water, under the hydrothermal fluid. And with respect to this research, we wanted laminated samples. Unfortunately, we do have laminated samples. The bottom image is an image of a silica deposit from a hydrothermal vein inside a mine. It too is composed down to the trace level of silica. So the coloration differences within that sample are the trace element level. Why do we choose Geyserites though? Geyserites form, as I said, at the high temperature end of the hot spring system. And evidence shows that life on Earth arose around hydrothermal systems. So that was one reason. Also, early life is chemoautotroph. Photosynthesis had not evolved yet at that time. And the microorganisms in the Geyserite samples are chemoautotrophs. So that's plug number 2 for choosing Geyserites to start with. Also, the microorganisms are very small. Due to the environment they're in, they're not able to develop large microbial mats, which then take on characteristic microbial mat architectures, which you find further downstream or down the hydrothermal gradient. So that was another reason why we wanted to choose these. So with that, we move on to texture analysis. I do need to give you a brief highlights tour of the history. We only have 3 stops, not going to be too painful. The first one, we start way back in 1973 when Robert Herlick and his colleagues set out to not only define but characterize what human vision picks up on so we can identify the differences that we see. After they determined the characteristics of vision, they then developed equations to quantify human vision and what allows us to see the differences that we see, for example, in the carpet that we have in this room, or in the image of the red leaves that I have up there on the screen. So now we're going to advance all the way up to 1997, and the National Institutes of Health released open source image processing software called ImageJ. Well, this in and of itself is not exactly maybe considered a highlight. It is important for our next step in the timeline and the last one, which is 2003, when Julio Cabrera released a GLCM, or the gray level co-occurrence matrix macro that works with the ImageJ software. So now we need to dive into the details. Why texture analysis, and what questions are we trying to answer with this approach? The 2 questions that we're trying to answer is, first off, does texture analysis, which I will get into more details, don't worry, you'll find out a little more about it. But texture analysis, we wanted to know, can it distinguish the 3 different types of samples that we selected for this initial study? Is it going to clump them all together, or will we get 3 distinct samples? Then the next thing is, can it tell the difference between the different components that comprise a lamina? And I'll also get more into that. So with that, for texture analysis, this is an image that I took with a stereo microscope. Take that of a region of your interest. Then I clip out sub-images is what I call them, which are represented by those white square boxes up there. Those are 32 by 32 pixel sub-images that were cut out. After that, the sub-images need to be converted into grayscale. I know it doesn't show very well on this screen. It doesn't show up very well on my computer that I've actually converted it to gray level, but it is converted to grayscale. There are 2 caveats with working with the macro for texture analysis. The one is it has to be a square. That's based in part just because it's a matrix. Then the second one is the macro that Julio wrote for texture analysis that is based entirely on the 1973 work done by Herlich and their equations. So that's why it has to be grayscale, because computers didn't handle color, and we were just past back in 1973 having to use punch cards to program computers, you know, so fortunately we've advanced a little bit since then. Okay, so what next? Now we get to go to the macro and actually start using the texture analysis, or the GLCM is what it comes up in if you install this macro on your computer in the ImageJ software. I primarily put this up here so you can see the variables that Herlich and his colleagues identified for looking at human vision. I am not going to bore you with the details of this. It would take the rest of the session to go through that. So after you input what you want it to look at, your step size, you can choose different pixels that you want. I always choose 1 because there are calculations that involve the nearest neighbor, and so I choose a step of 1, just that way I don't introduce any artifacts into the data. You click OK, and you get a nice pop-up window with these values that I still have yet to figure out how they correlate to what is in the subimage, but the values do actually mean something. I just haven't figured out if a high value in one subimage is a low value in another. I don't know why it does that exactly, but put that into a spreadsheet after you go through all your subimages and all the rotations of your subimage. You get these huge spreadsheets and start performing statistics. Our first quick and dirty test that we did just to find out what results are we going to get? You know, are we going to be able to see the different samples? Are they going to get separated out statistically? So we did a hierarchical cluster analysis, and they separated out fairly nicely. At the top, in the red, as the legend says, we have the spicular Geyserite. We have one anomalous little green data point that made its way into the subaqueous Geyserite, and then we have our control, the hydrothermal vein infill. We were pretty excited at these results because it showed that it's separated out of 3 samples really nicely. So we wanted to take it up a level and put all the data points into a k-means cluster analysis, which is a little more complicated because it takes all the data, it plugs it in and plots it, and then the algorithm is an iterative approach, and it tries to cluster the data based on means and distance from the centroid value that it comes up with. And we did get fairly good separation on the spicular Geyserite, but our subaqueous and our control sample, they didn't get resolved out very well. And my thoughts on this, I have yet to talk to my advisors about it, but my thoughts is they formed in similar environments. The hydrothermal vein infill, it's a fluidic environment. The subaqueous, a fluidic environment. So I can see the similarities, you know, where they didn't get flushed out quite as nicely. So now we get back to what was it that we were actually, or what were the subimages that I collected? What was I looking at? If we break it down into the anatomy of the lamina, I discovered that I didn't collect all the data points that I really should have on this run of data. I collected the abiotic, which is a massive dumping event of silica. And so you get these thick silica horizons. Then, and I did this for illustrative purposes. This is not representative of the high temperature end of the system. I chose to use green photosynthetic, like filamentous type microorganisms for illustrative purposes. Like I said, this doesn't represent the high temp end, but you have a biotic part of the lamina. And then we have these 2 interfaces where we have an abiotic surface where we had that massive dumping event of silica. That surface then has to be colonized by microorganisms. They thrive, they flourish, they're living life, and then until they're snuffed out by another abiotic deposition. So these 2 interfaces, they're going to be a little bit different. For this study, I only captured the abiotic to the biotic transition. We then thought, what if we do the k-means analysis looking at each part of the lamina? So we plug in the abiotic data points only, run the k-means, they got clumped together rather nicely. Same for the biotic. And then the one interface that I did. So what I found out from one of my advisors is that sometimes with statistical methods, you can get some conflicting results. But when you figure out what's going on in your system and what the statistics that you ran actually are doing, you can figure out where the mistakes occur. So we did end up being able to distinguish the different parts of the lamina, and it also clumped them out distinctly based on sample type as well. So why are we doing this? What is the goal? You know, where are we headed? Obviously, I will be incorporating additional samples from different environments with the goal of developing a computer program that, when coupled with spatially correlated chemical data, can help determine whether or not a laminated sedimentary structure has been influenced by microorganisms or not. And with that, I leave you with the summary of our findings that with the combination of texture analysis and statistical methods, we were able to distinguish between the different sample types and the different lamina components. So. Oh, we'll get back to you.
Hello. So I had a quick question back on your clustering slide, if you
Sure.
mind going there real quick.
Yeah. Which one?
The very first tree you had.
Sure.
Yeah. So looking at the tree structure, first and foremost, what are the support values for your nodes, if
Good
you have
question.
any?
I don't have that information.
Okay. Secondly, when you look at the topology of this tree, I noticed that the top group of your subaqueous Geyserites are actually grouping with the rest of your spicular Geyserites underneath the very top approximately 6 or 7 lineages.
Yes, I did not mention those. My apologies.
Okay, so what's going on with those? Why are they different?
They're similar. When you, at higher magnification, when you look at the massive dumping of silica in the spicular Geyserite, there are also massive dumpings of silica in the subaqueous, and texturally they can look very similar.
Okay, thank you.
Sure.
Thank you for your talk. Have you looked at any of the chemical profiles yet, utilizing something like Raman or MS, like mass spec work? Have you correlated your work yet with any of the other studies on your stromatolites?
Not on these, I've not, but I have done some EDS work, which I know is not quantitative, but you do get carbon signatures that will come out. They will plot distinctly, as well as the silica, and then there are other trace elements. I did find in some different work that I did with filamentous microorganisms further down in the hot spring system, there were salt deposits around the filaments, and so there are trace elements in there. We have to use higher resolution methods to resolve that better. Good question, thank you.
Any other questions? I guess if not, then let's thank Shana one more time. And
Thank you.